Hi, everyone. Welcome to the Johns Hopkins Cerebellum seminars. Today, as our guest, we have Haining Zhong. Haining received his degrees in biology and electronics and computer technology from Tsinghua University. Then he came to America. He did his Ph.D. here at Johns Hopkins in neuroscience with King-Wai Yau, and then did postdoctoral training with Karel Svoboda and Eric Betzig at Cold Spring Harbor, then at Janelia Research Campus. In 2010, so about 15 years ago, he started his independent lab at the Vollum Institute in Portland, Oregon. He's currently a full professor and senior scientist there. His lab develops imaging technologies and uses these technologies to study the cellular mechanisms underlying locomotion, learning, decision-making, and he's developed strategies based on mouse genetics and on CRISPR-based gene editing to fluorescently label endogenous proteins for imaging. So his lab has been able to image neuromodulatory signals and in cellular resolution in behaving animals during both locomotion and decision making. And today we're going to hear about his work on the cerebellum.
Okay. Thank you, Reza, for the organizing this seminar series, and thank you for the opportunities to speak here. Like Reza said, I did my PhD at Hopkins at PCTB, and then really it's always fun to be able to give a talk kind of to the Hopkins audience. So overall, my lab used microscopy methods to study the neuronal structure and signaling mechanisms at different scales, individual synapse, individual neuron, as well as at the systems level. And today I will really talk about a new talk in my lab, and also for the first time, that about the cerebellum, and then particularly about the cerebellum function in decision. And so I really welcome a lot of feedbacks, and then so please feel free to interrupt me kind of for if you have any questions, and I think that will really help me to learn the field and so on. So overall, my lab is have a technology development side and a biology side. So if I can advance, right? Sorry, somehow. Sorry, somehow I cannot advance my. Oh, okay. Now it works. Okay. So one thing my lab has been focused on in the past few years is to take all this kind of image, all these different second messenger pathways that's downstream of GPCI and further downstream from all these important neuromodulators in the brain. And then our standard is always we want to be able to image these signaling pathways in vivo during animal behavior. I'm glad to say that over the past few years, we have been able to do so for a number of these, and then for the remaining ones that we haven't been able to do, we have things in the pipeline. So by the same time, we also use our technology to study biology, such mostly animal locomotion, but we also start getting to decision making. But all of our study has been in the cortex, also in the striatum. So cerebellum, it's a new adventure for us, and in part largely because of this student who just graduated very recently, that a few years ago, he decided to join my lab, and then but at the same time he has a prior training from Mike Hauser's lab that many of you probably know that is also one of the kind of figures in the cerebellum field. He said, "Oh, I like your lab, but at the same time I also want to keep working on cerebellum." So then I say why not just have fun there. So then really what I talk about today is largely his thesis work. So a brief outline of what I'm going to tell you today, that so first we start by imaging the dendritic calcium of Purkinje cells, which largely reflect the climbing fiber activity, and particularly we have a study about the decision information carried in this climbing fiber inputs. Then once we and after that then we ask this basic question that like can cerebellum even directly drive decision input if it receive information about decision? Then at the end, like if I still have time, I will talk a little bit about my home base, kind of the new modulation of the cerebellum that we just starting, but the result is quite exciting for us. So I won't say like not all of the talk that I'm talk about today are unpublished. So I really hope to get feedback. So we all know that like cerebellum has a kind of most people know cerebellum is important for fine movement control. But at the same time, evidence also start to accumulate that cerebellum can also involve in cognitive functions, such as in this study now, actually almost 30 years ago now, that in an fMRI study, they asked a person to, sorry, like I have some jitter in my end, but basically like they asked the person to handle a few balls and then like so, but by this like you see very little activity in the cerebellum. But if at the same time, in addition to handling the balls, you also ask the patient to try to sense the texture of the ball. There are two type of ball there, and all of a sudden now you see like a robust activity in the deep cerebellum nuclei as reviewed by fMRI study. So then in the clinical side, like there were also these children there who unfortunately like has kind of injury kind of in their cerebellum, or sometimes due to various reasons they have to have surgery and cut out part of the cerebellum. Then if you have one of the kids like to copy a figure, she can do it, so the motor function is fine. However, like if you ask her to immediately record, like the picture start to become disintegrated, and then after 20 minutes, it becomes something like this. All this suggests that like the cerebellum, in addition to the classic motor function, may also play roles in cognition. And so and this and all this like kind of at the experimental kind of in mouse studies like become one of the recent focus such that like as what we show here is the big classic basic structure of the cerebellum kind of circuit that Purkinje cells, the principal cells in the cerebellum cortex, receive input from granule cells and also climbing fiber. So historically, it's thought that like the granule cells carries sensory, motor, and contextual information to drive Purkinje cell activity. But at the same time, it also receive this motor arrow and timing feedback signal from the climbing fiber that drive the learning, basically like to pairing to control how the granule cells can control Purkinje cell firing action potentials. But one of the kind of recent landmark study that turning people's attention is that like Lynch lab like published a paper a few years back, like that shows that like in the granule cells like there's you can also encode reward expectation. Then in parallel, like in Mike Hauser and also Karel's lab, they show that like the climbing fiber can encode another type of feedback signal, the reward prediction arrow. And then so like this basically suggests that like the cerebellum can also use this information to study kind of to compute reward related behavior. So and my lab like has been focused on the climbing fiber input. So I will elaborate a little bit more on the reward prediction arrow that I'm sure most people know, but just in case for some of the kind of newer students, that like the reward prediction arrow was classically kind of described in dopaminergic neuron in the midbrain. That like if the animal all of a sudden receive a reward, so like this is beyond its expectation, so there's an arrow there. So you have the dopamine neuron action potential response. This study was in monkey, but now has been replicated in many animals. But then if you pair the reward with a conditioned stimulus, now the saliency, it comes from the conditional stimulus. It already signaled the reward. So then at that point, like the dopaminergic neuron will fire. Then when the reward truly comes, since the animal already expected, so there's no error there, and then you actually don't have a response there. So like this kind of basically is a characteristic of the reward prediction error. We often abbreviate as RPE. So then people start to realize that like the climbing fiber can carry RPEs, such as in this study that like there's a visual kind of reward paradigm, and then like the author has been measuring like the different location in the cerebellum, simplex, cruise 1, cruise 2. Those are thought to be kind of more related to kind of non-motor functions of the cerebellum. What they found was that like in both the simplex and cruise 2, they see this signal very similar to RPE in that like before the animal learns, like when reward comes, you see this response of the in the dendritic calcium of Purkinje cells, again reflecting the climbing fiber inputs. But after the animal learned that the cue can indicate reward, the response move forward, and then now it responds more to the cue and less to the reward itself. So this is consistent with the classical RPE. But they also report at that time that if the image cruise 1, which also thought to be potentially related to non-motor function. However, like in both cases, it seems to respond to the cue and then not so much to the reward. Then it raises the question like, but this is not RPE, so right? And then, so it raises the question like, what kind of information it carries. So, so like this was 2019, and then in 2020, like a team started in my lab. So then we discuss and we say, oh, maybe we can start by examining like what kind of information the cruise 1 climbing fiber carries. So in order to ask the question, team designed a multi-stage auditory go-no-go task, and that also built from his background as well. So he will first like use either 8 kilohertz tone and 14 kilohertz tone to signal reward, and then followed by a trace period, then the reward will either come or not come. First, in the generalization stage, that both tone will be paired with reward, and then naturally the animal will lick, basically like a kind of in response to both tones, and then so it's color coded. The yellow means response to 14 kilohertz tone, and purple correspond to 8 kilohertz tone. Then after animal learn the generalization stage, we go to a second stage in which the 14 kilohertz tone is no longer signal reward. It will signal kind of no reward, and if the animal lick, actually it will it's a false alarm. The animal will receive a small punishment by timeout for a few seconds. So and the animal quickly learned that like now it mostly only lick kind of in the presence of the 8 kilohertz tone, but not so much in the 14 kilohertz tone. And the data is quantified here that in the generation phase, like both animal does not distinguish the tone, and in both cases it will lick a lot. However, during the discrimination, it lick during the 8 kilohertz tone, but in the 14 kilohertz tone, when it hear the sound, it will try to lick. However, it will try to suppress it to avoid timeout very quick. And as evaluate in the field, I'm sure like most of you know that we evaluate the animal behavior using the D prime index, which is the comparison between the 8 kilohertz performance and 14 kilohertz performance. And usually in the matter of 3-4 days, the animal can cross the threshold of D prime of 2 and then become an expert. So from here, like we the first question we ask, and in fact, like when team present this in the lab meeting, so the people in the lab ask, like, does cerebellum even has anything to do with this behavior, right? So to address that, like we use a channel adoption kind of optogenetic strategy that we breed this PCP2 Cree mouse, which express Cree only in Purkinje cells, and cross it with AI32, which express channel adoption in a Cree dependent manner. So then what this allow us is to express channel rhodopsin in all the Purkinje cells. Then we open the window only up on top of closed one, and we use a light to shorten it. And as we know that these Purkinje cells, they will suppress the cerebellar nuclei. Therefore, like you excite Purkinje cells, we will suppress the cerebellum output to the other part of the brain. When we do so, like if we stimulate the Purkinje cells in both the Q and also the trace period, like the animal performance goes down. If we do it in the outcome, our outcome period, at that point, like the animal already decide to lick, and then like there wasn't any difference. So so like this about Purkinje cells, but Purkinje cells, as we know, like can fire complex spikes from the climbing fiber input, but it can also fire simple spikes. So like which one like is important? So here like we further do this experiment that like we inject channel rhodopsin to inferior olive, which give rise to the climbing fiber in the cerebellum. And this shows that like we can successfully like label this cerebellum using the M-cherry fluorescence, kind of that is tagged to the channel rhodopsin. And then what we found is that like if we give optogenetic stimulation during the Q and trace period, and again the animal performance kind of become deteriorated. So all this suggests that like the climbing fiber input to the cerebellum is important for this behavior task. Then we ask like what's happening like to the signal? What kind of signal is carried in this climbing fiber input? And again, like as I mentioned earlier, that like we use two-prong imaging, particularly the GCam AS, although it's supposed to be the slow variant of the GCam, but it's also known that the AS, since they are so good that it's very comparable to 6F in terms of its kinetics, that many people use in imaging these dendrites. And by imaging the dendrite, by imaging the dendrite, like it mostly detects only the climbing fiber inputs, the calcium activity. This shows our raw data and by using SWI2P to analyze it, we can extract individual ROIs. This gives us the calcium trace of individual dendrites. Then from there, like we further perform our analysis based on this extracted data. So first, just like what many other people do is that like we just combine like all these ROIs together, and we ask like whether go from the generation to the discrimination phase, whether there's any difference. So this shows the 8 kilohertz tone response. The 8 kilohertz tone like signals both kind of reward both in the generation and discrimination phase, and we see the response become a little bit stronger during discrimination, although not that significant, but it become a little bit stronger. However, like when we go to the 14 kilohertz tone, now to 14 kilohertz tone in the discrimination phase actually means that there's no reward, although in the generation phase it also signals reward. So if by the traditional RPE model, one would expect that like the signal during the Q and also trace period should go down, but instead like we actually see it also goes up. In fact, like if you overlay the 14 kilohertz response in the discriminant phase with that of the 8 kilohertz response in the discriminant phase, one see almost no difference other than in the outcome period. Like I think the CRISPR ones also still has a movement component. So then like in the outcome phase, like there's a little bit difference. So then we ask but since like it doesn't seem to be RP, we ask maybe what kind of information like it may still carry, and whether we ask whether it can contribute, whether it can use the information, the calcium information carrying this can that be measured to decode what kind of tone the animal is listening to.
Haining, may I just ask you a question?
Yeah.
Just to clarify, sure. In the last slide that you had, so the generalization phase was the phase when both cues gave reward.
Yeah.
and the discrimination was only one of them give. And the results here are that there's a slight increase in activity.
Yeah, in both cases.
For both of them.
Yeah, so like that's what we see, and then that's how we feel that it's not consistent with the classical RPE model. So then we decide to use this calcium activity to train a model. Here we use this support vector machine that Tim learn kind of from kind of his past experience. So that like we use 80 randomly select 80% of the tries like for training, and then we save 20% like for testing the model. And then after training, what we found is that actually the calcium signal in the discrimination phase was sufficient to train the model to decode kind of whether the animal is listening to 8 kilohertz versus 14 kilohertz. And versus like this versus as control using the information from the generalization phase, like the machine failed to decode like the what kind of tone it is. At the level, and this there's a component of population coding that at the level of individual neuron, actually like it's the ability is relatively moderate. However, if you include more and more ROIs within one field of view, you can decode better and better. And also, of course, if you shuffle the data, you can no longer decode it. Then from here, like we ask why and then like how this happened. For that, like we convert to a kind of a more analog analysis using the principal component analysis, which basically like here it shows a plot of the first three principal components that explain more than 80% of variance in our data. And here shows the time-dependent trajectory of the kind of dendritic calcium activity. So and we found that like in the generation phase between the 8 kilohertz or 14 kilohertz, the neuronal trajectory was very similar. However, when we go to the discrimination phase, things have become significantly different. That like you can see like the between the 14 kilohertz and 8 kilohertz, particularly the correct rejection of 14 kilohertz, like the neuronal trajectory kind of projects kind of very different. First, they go somewhat similar towards or towards the upper right, but after about 300 milliseconds, it starts to diverge. The interesting thing is that like we also had this as a fraction of this false alarm tries. That like it's a 14 kilohertz tone, so there's no reward, but the animal somehow saw that there is a reward and also keep leaking similar to in the when the 8 kilohertz is presented. So it also kind of diverge at about the same point, but it diverged to a space that like that is somewhere in between of hit and correct rejections. So and this slide shows a couple of these important control experiment to suggest that like this neural trajectory is more about the decision rather than about the movement. As we know that cerebellum, any part of cerebellum potentially can be related to movement control. But at least in this assay, so we take the we separate the animal leaking like by its onset, and we take the first quadrant or the last quadrant, so they are quite different. However, the neuronal, the dendritic calcium activity goes a very similar trajectory in this principal component and kind of space. And similarly, that like when we look at the animals kind of tries with a low leak rate versus with high leak rate, the neuronal trajectory also goes a very similar trajectory. This suggests that like the divergence that we saw in the previous slide was probably more related to animal kind of decision or choice or action rather than the movement per se.
Sorry, can I ask a question about this slide?
Yeah, sure.
I was wondering, it looks like you're doing this analysis on your delta F over F data, right?
Yes. Yeah, Z-score. We actually did Z-score.
Okay, yeah. So your Z-scored fluorescence, and so this is reflecting the kind of tail of the calcium transient of the GCamp response, right? I
Yeah.
wonder, what you think is actually happening during this later period. Do you think that, like, how would this look if you had done deconvolution or kind of spike extraction, or done this with eFIS, where you actually have the timing of the complex spikes? Like, do you think that this is the same complex spike, which is broader or larger, that is encoding these differences, or do you think that there's a kind of second wave, half a second later, or whatever, of activity that is independent of the kind of sensory transient you see at time zero? Do you have any sense of that?
Yeah. So like we actually look at that. I think like sorry, I think I now with you ask this question, I realized that I did not put that slide in for this talk. However, like we did try to deconvolute and then get the complex spike identity, and then also look at the waveform. We didn't see any significant change in the waveform, and appear that like the difference is majorly comes from what you say that a second wave of complex spikes after the initial wave. So we think that there's an initial wave of spike that was more responding to the sensory saliency kind of per se. Then there is a second wave of complex spike that is that signals the decision. So like this data actually you can find in our bioarchival preprint. Like yeah. So sorry that this is a very important point, and really thank you for asking it, and I think like I made a mistake not putting it here. Yeah, so then we asked like so always like this is still a populational analysis. So it tells us that like the after 300 milliseconds, the animals start to kind of evaluate the signal and then start to kind of the signal start to diverge. Then we ask like at the individual dendritic level, what is happening? So to do this, like we go to the decoding kind of machine, like that I showed you earlier, but now we look at the individual dendrites. What I see is that like in the generation state, like there's you can see the average of the decoding score is very close to 0, and then the x-axis is a difference between the fluorescence between the 8 kilohertz tone versus 14 kilohertz tone, and also on average like it's 0, and then there are a few outliers, like the threshold is about 2 standard deviation kind of the variance, and then so there were a few outliers, but relative there were much kind of relative few of them. But when we go to the discrimination phase, where the difference between A versus 14 kilohertz tone kind of become important, all of a sudden we see a lot more of these outliers, and then also these ones tend to have better decoding scores. And the interesting thing is that like they can really go in both directions. On the right is the ones that has a bigger response in 8 kilohertz compared to 14 kilohertz, and then on the left is a more bigger response in 14 kilohertz than 8 kilohertz. If you just average everything, like the effect is very small. However, like when you look at this individual RIs, the effect become much more obvious. Really says that tells us that we cannot only look at kind of an average response of all dendrites.
Quick question, Haining.
Yeah.
So all this analysis is done on the Q window or also the trace window?
This one, I think it's mostly, I think like it's the trace window. I think like it should be after the. I think like it's the trace window. I need to verify with my student, but there's a chance that he may start from 300 milliseconds when the signal start to diverge. But mostly it's the response in the trace window before the outcome period.
And after the Q. So we don't think this is a sensory related response.
No. Yeah.
Right. Thanks.
So then this shows kind of the some of the individual RI traces like here, and you can see that like in some cells like the 8 kilohertz response is larger than the 14 kilohertz correct rejection trace, and then some kind of the vice versa. And the divergence, the initial phase actually really is very similar. The divergence start to happen at about 300 milliseconds after. Then we take advantage of our data. Then we realize that we can further divide, further dissect what kind of information the neuron carries. that like we take advantage of this false alarm tries. You see, this shows the leaking response. The animal in the false alarm tries, although it's present to a tone that signal no reward, but they really somehow think that there may be reward, and then they leak in a way that is very similar to the hit tries until in the outcome period that when there's no reward, and then the signal when the animal only read that there's truly no reward, and then it start to kind of stop leaking. And when we do this, like we find that like many of these cells can be fall into two different classes. In some of these cells, the false alarm response is very similar to the hit tries, the 8 kilohertz tries. We call them the action choice because like in both cases, like it reflects the animal is going to leak or not leak. So, but in the others, in the others, like the false alarm kind of trace, like it's more similar to the correct rejections. So in this case, like it's more similar. I think we think that like this dendrites, like their signal kind of reflects the Q identity, whether it's hearing really a 14 kilohertz tone versus 8 kilohertz tones. So, but overall, like we think that like a subset of this climbing fiber input can either encode the choice or maybe the Q identity. But there are also many other ROIs like that doesn't do much kind of in this context. So then from there, like we ask one more question as all experts know, like in this field that the microzone is a big thing, like in the cerebellum, right? Like all these microzone are like all these different cerebellum functions are sort of organized into microzones with at the order of 200-300 microns, and we found that basically like the difference between the 8 versus 14 kilohertz kind of appear to kind of form this patch that is consistent with the microzones. So like this, you really need to look at the difference rather than just look at its own activity. If you just look at the climbing fiber activity without doing the subtraction, like it's less obvious. But now once we in the difference, we see this kind of continuous patch, and even with our relatively small field view, like we can see this boundary between these two different microzones. And in this nearest neighbor analysis, like of course, like the ones that share the ones to a nearest ROI to the same response, it's much closer compared to another ROI that give a different, the opposite type of a response. But we also realize that there may be sub-organization structure within one microzone. So that's when we try to map the choice ROIs and Q ROIs as we discussed in the previous slides, we found them the best way to describe them is that they must be intermingled with one another within one microzone. And also in this nearest neighbor analysis, like you no longer have a difference between the same type of kind of ROI versus the different type of ROI. So we think that like so, and right now like we don't quite have the capability to further go on in this direction, but we feel that if people want to study how the cerebellum function, like considering all this submicrozone organization, will also be an important component of the analysis in the future.
Could you clarify? Just a quick question.
Yeah, sure.
Could you clarify if the tone preference was before or after training?
The tone preference, like this is after training.
Do you think it requires training?
I think like it become more significant, more obvious, like after training, but you can use the. This is a very good question. I think like we need to go back to do more analysis before I can answer you with good confidence. But
Okay,
it's a very good question. But even what we know is that like even with kind of baseline activity, even without kind of the tone stimulation, basically like in those inter-trial intervals, and then you can already separate the same microzones. Although like you don't know like exactly what they're responding to, but then when there's a tone response, then you can see this further microzone kind of respond to one polarity than the other.
One thought that I might add regarding this is that it's not just a tone preference, right? Because it's also two different actions. So in the case of 8 hertz, you will lick. In the case of 14 hertz, you will suppress the licking, and both are things that you have to control. And in the one case, not to do something is just as important as doing, because if you did it, you get punished for it.
Yeah. That's great. Yeah, that's a good point. Yeah, but I think like yeah, sorry.
Sorry. Go ahead.
I guess, sorry. So I guess part of my question is that there's nothing special per se about 8 and 14 kilohertz, but it does seem like there's such a nice clear boundary here. So that I guess is part of why my question arises, in that it only arises because of learning. And if you had like 3 and 10 kilohertz, you'd then see maybe a similar boundary.
That's what we think. The reason we pick 6 and 14 is because they have the mouse is supposed to have comparable sensitivity between these two frequencies. And then like we actually start with 4 and 10, but then there was a postdoc in the lab like who has previous auditory kind of research experience, kind of made a big deal out of it that the mouse cannot hear 4 kilohertz. Yeah, so that was why. But yeah, but we kind of think that like our thought is that like the microzones are probably already there, and then like during the learning like it will be recruit to to process or represent kind of one type of difference versus another. So that's generally what we saw. But I think like we should go back and then verify that.
Yeah, so kind of a question about the intersection of these two things. So if we think like the map you're showing on the left is representing probably, the microzonal modularity. Do you have a sense of whether the kind of choice in Q encoding, like how it actually follows these microzonal maps? So it could be, to be specific, for example, one could imagine choice, like there being choice information on either side of a microzonal boundary, but say the encoding might be in the opposite direction or something like that. So we saw this a lot with reward responses. You'd see on a microzonal boundary, on one side, the cells are positively modulated by the reward, and on the opposite side, cells are negatively modulated by the reward. So do you know whether here on the right in this analysis, the choice and the Q information are actually represented in the same way across these boundaries that you can probably identify based on your tone preference responses?
It's a very good question. I think like it's something that like we haven't been able to address due to technical kind of capability, because my lab has been mostly using Galvo, because we usually do lifetime imaging, which is a little bit slower, so we don't need resonance scanner. And then, but at the same time, in order to image calcium kind of at relative fast speed, we have to go to relative small field view so that it can scan faster. So this limit our size that like we are usually at about the size of one microzone size, and then sometimes we can see this boundary, but we can never cover 3 microzones simultaneously. So I think like it's a very good question, but we don't have the answer right now. But I think like now we already acquire a resonance scanner in the lab, so I think that's something that we can address in the future. Okay, so yeah, so then we the next question we ask is whether there might be learning, right? Like how this information is encoded when the animal learn. To do the learning, we perform a reversal, a kind of a task that like now after the animal already know that like 8 kilohertz kind of is associated with the reward. Now we change it such that 14 kilohertz is associated with reward and 8 kilohertz is associated with no reward. and the animal can learn. It takes them a few days, but eventually it can still get to a D prime of 2 or better. So and in this principal kind of component space, we found that like there's an interesting switch. Like now for the 8 kilohertz, now it like now instead of going downwards, originally it now start to go upwards, and then the heat basically like maintains priority, but in terms of tone, it start to go down, and the false alarm remain to be kind of somewhere in the middle. And so like then we ask like at the level of individual ROI really what is happening. And I'm sure like many of you have this experience that like it's not exactly easy like in this field to try to match ROI from day to day and from kind of it's really like from kind of sessions to sessions. But then the team basically take this heroic effort that he manually kind of score the dendrite, and then like he was able to find a subset of the ROI that he feel with a good confidence that he can match across days. Then we actually recruit a second postdoc who is not in the project, and then verify like his scoring, and then make sure that it's reasonable. And then so then like he had this about a quarter of them he can match across 3 days. This shows one of the example dendrite that like in the generalization phase, none of them has much response. Then during the discrimination, it has a bigger response, particularly the 8 kilohertz has a larger response, but then it switch during the reversal. And overall, like what we find that if we saw all the cells based on the delta response between 8 and 14 kilohertz in the trace period, and we found that like and then we look back at the generation phase, there's a very little structure. However, like in the reversal stage, it seems that like the ones that has a stronger 8 kilohertz response tend to have stronger 14 kilohertz response now, although there's also significant noise there. So overall, like you can see this very loose but still statistic significant relationship between the response the response difference in the discrimination phase versus the reversal phase. So I think like what all this suggests to us is that like at least for the same tone, like the response has to basically like go to different Purkinje cells in order to mediate the behavior. So we think that like the input to the climbing fiber input to the Purkinje cells is flexible in that sense. So therefore, like a little brief summary of what I tell you this far, that we think that the climbing fiber input includes one is important for decision execution, and particularly that like there are a subset of climbing fiber input encodes either the perceptual or action choice or movement planning, but something along that lines. And then there are other RIs that kind of encodes the Q identity in the climbing fiber, and this information supposedly should be important for the animal decision execution. So now when we realize after this first study, this relative complete, then the team start to think that, oh, like since they carry this decision information, maybe we should ask like whether cerebellum can even directly drive decision itself, right? So and so like then in some very preliminary result, like I'll show you some of the ongoing work. So and to ask this question, like we use an optogenetic approach, and then you must know that like other than this study, there are many other studies as well that pure using optogenetic stimulation in the cortex or in many other brain regions, like you can use it to train the animal to kind of for decision behavior, and particularly this Ren Eli paper show that you in the sensory cortex you excite about 50 neurons, and then you'll be able to reliably drive the animal behavior. But there's we didn't find similar studies in the cerebellum, so then we decided to do it ourselves. And again, like we focus on cruise one, we use this PCP2 Cree AF32 mouse in which Purkinje cells already express channel rhodopsin, then we shine line, and we pair the tone 8 kHz. If we pair with optogenetic stimulation, it means reward. If the tone alone means no reward. The animal can actually learn this very readily, and then you see like early in the phase, the animal will link to both conditions. However, very soon, like you can see like the purple dots are mostly gone. This is because when you the animal will only lick when there is a kind of optogenetic stimulation paired with the tone. We usually do about 100 tries a day, and then even within the same day, the animal can learn, and then the animal can become an expert within 3 to 4 days. This shows a couple of control experiments, but it's important that first, like if you give a different gradient, different intensity of light, you see this graded animal performance, suggesting that like the animal, that the Purkinje cell response like is somewhat kind of in an analog mode, rather than an all non mode, that you may need to recruit either stronger Purkinje cell activity or maybe more Purkinje cells in order to get kind of more robust decision. And then also, if we move the fiber a little bit off the window, so the fiber will still shine light, so then whatever light leak out that's sensed by the animal light will still be seen. However, like the performance goes to 0. This suggests that this really requires a direct excitation of the channel rhodopsin cells. So then from here, like and what we have found so far is that like after the training, that you actually don't need the 8 kHz kind of tone at all. Like if you give the animal 8 kHz tone, the animal won't lick. However, if you give the optogenetic stimulation alone, the animal can lick almost 100% of the time, with a little bit difference in that the response seems to be delayed. This suggests that like some of this photo activation kind of like will drive kind of a downstream effect and then further drive the animal decision. But still, I think like the Purkinje cells is likely to be part of the decision circuit, or certainly can affect the decision circuit there. And one last thing is along these lines, we asked whether the animal can reverse like the learning. This is because like when we excite the Purkinje cells, like maybe it can, one alternate hypothesis is that like maybe the Purkinje cell can only drive leaking, right? Like instead of drive the suppression of leaking. But here, like in the reversal, we found that the Purkinje cell activity can also drive the suppression of leaking, that now after the training, the animal kind of only leak in the absence of Purkinje cell stimulation. And also it takes the animal a little bit longer to reverse it, but still over 5-6 days, some of the animal can become expert. So yeah, a brief summary of this part is that we think that the Purkinje cell activity can directly tap into the decision-making circuit. It may not be direct making decision, however, it can influence how the decision is made, and then also the behavior association of Purkinje cell, whether it's a leak or not leak, harmful, it's flexible. The same suppression of some excitation of Purkinje activity can drive both leaking behavior and can drive kind of suppression of leaking behavior. So in the last a few minutes, like I will quickly show you some of our kind of very preliminary result. Again, like our home base about the neuromodulation regulation of brain circuit, and particularly applied to the cerebellum. So I have shown you like in the very beginning that my lab focused on taking all these intracellular signaling pathways for their imaging in vivo during animal behavior. And one pathway we have focused on is the PKA pathway. Upstream, they receive a lot of neuromodulator inputs and activity as well. And downstream, they regulate ion channels, neurotransmitter receptors, intracellular proteins, transcription factors, and touch upon almost every aspect of neuronal function. And particularly for my lab's interest, like we really focus on this pathway seems to directly control the neuronal excitability, which controls how many action potential a neuron can get under the same input, and which always like it's a circuit gain, and then it can have a major effect in the circuit function. So this shows an early in vitro evidence that like if you activate this pathway by give the new modulator norepinephrine in this hippocampal C1 neurons, norepinephrine increase PKA activity, and under the same condition, you get a lot more action potentials. So and we image the PK using a FRET-based sensor. For the sake of time, I won't touch upon this, but it was developed by Jin Zhang when she was still in Roger Chan's lab, and I mentioned her because for about 10 years she was also a faculty at Johns Hopkins as well, although she's now at UCSD now. Then particularly like we measure the FRET. I won't go into the mechanism, but it's hopefully it's clean enough. But particularly we measure FRET using a technique called lifetime imaging, which measure the time difference between the flow excitation to time to photon emission at the order of a few nanoseconds. If you align all these events, like the photon excitation event, what you get is a single fluorescent decay. The time constant of this fluorescent decay corresponds to a lifetime. But once it's plotted, the advantage kind of becomes obvious that in vivo in the brain, like there are a lot of things that change the intensity. That's why you can never become be super quantitative for intensity imaging. The different expression level can change things. Light scattering can change things. Drifting or animal movement and even heartbeat can change the intensity. But if you measure in the time domain, like if you normalize all these traits with a different kind of intensity, like the time decay is about the same. So this made a lot more stable and robust. In one the example that like we show like a few years back that for the same sensor, like with a lifetime imaging, you go deeper and deeper into the brain tissue, you get stable recording. However, even if you try to cancel out some of the other effects using ratiometric imaging, you go deeper, like the value just keep changing there because the brain tissue will scatter light. It will scatter different wavelengths light differently. Overall, lifetime imaging has a lot of advantages. And so then my lab, like over the years, we have tried to optimize like the ACAR sensor, the PK sensor, like for its lifetime performance. This shows, and then in the beginning, it was just really impossible to go in vivo, but at about 2017-18, we eventually crossed a threshold that like the sensor become good enough and combined with the lifetime imaging allow us to measure PK activity in vivo during animal behavior. Now we can even do this in free moving animal now. So, but like when the animal here shows the animal runs under head fixed condition and in the motor cortex, you see these neurons, they are somewhat similar to one another, but when the animal runs, only one shows high peak activity, the other does not. We based on our hypothesis, we think that the one shows high peak activity correspond to that like a high neuronal kind of the neuron circuit is engaged in the current behavior. The other neuron doesn't show high peak activity, is in a standby mode right now, but it may become engaged in another animal behavior. But these are some of the $1 million questions that my lab is still trying to directly demonstrate. But now this is a kind of cortical data, then we try to apply this technology in vivo, and we get something very striking. That here, like we use a color scale to indicate peak activity. Red means a high peak activity, and in this scale, like the red means almost full activation peak activity. And we see this striking gradient of peak activity that there's almost nothing in the soma to almost full PK activation in the dendrite. That nobody just even expect, like we don't like we certainly didn't expect. And in the field of Purkinje cell biology, if you look into literature, PK was never a major thing. About more than 20 years ago, David Linden at Hopkins, also his former postdoc, kind of Chris Hansen, now in U Chicago, published a couple papers about PK may modulate excitability, but nobody realized that there's this striking PK activity there. But so then this really provoked us to further carry out the study. And first we did was to verify that the PK activity is indeed specific. We do this using by also expressing in this Purkinje cell the PKI. PKI is a naturally occurring protein that specifically inhibit PKA. If you overexpress it, you can kill all PK activity. That people have shown this in many cells. And when we express PK activity, the gradient was completely gone. This suggests that it's not some kind of non-specific kind of artifacts. And then we also take advantage of PKI that can suppress PK activity to ask the function of PKA. So many of you probably know that if you cut a acute brain size of Purkinje cell, you patch it, the cell will spontaneously fire at about 30 Hertz. So like we found that indeed this is the case if we don't express PKI in the neuron. However, in the cells that express PKI, on average, like there's very little spontaneous activity. Actually, the one we show here is one of the ones that are high responders. And then when we can also break into the cell, and then so they allow us to do current claim recording and inject currents. And what we found is that like on average, the cells, if it express PKI with the PK activity is suppressed, the excitability goes way down. You need to inject a lot more current in order to get the same number of action potentials. And also like for the cell that does not express PKI that is in the same brain slice, their excitability looks fine compared to kind of for some of the cells that we previously recall. So actually, when we say we, really I mean Skylar Jackman, like my colleague at the Vollum Institute, and also particularly his postdoc, Dennis. So then that they recall in the Y-type animal, kind of previously, the excitability was very comparable. Okay, so then we ask like whether the PK function is important for animal behavior as well. So then we inject PK across multiple region in the CRUS1, which is quite big. And then but when we do so, like we found that in this very simple tone triggered kind of a reward paradigm where the animal will lick kind of after the tone. And then when we extract PKI, it seems that the leaking is a delay, and then also it become a lot more variable, as quantified here that like for the PKI expressing animals, basically like both the reaction time and variability kind of greatly deteriorated. Also, like we still have a limited number of N, and we are trying to make this kind of more consolidated, but the result look pretty good so far. So what I have tell you for this last bit is that like we unexpectedly found that the PK signaling seem to play a very major role in regulating Purkinje cell function. Then always the next $1 million question is basically what controls this PK signaling. And it's known that like serotonin, dopamine, and also norepinephrine all project to the cerebellum. So this is our next question is we ask like really what the major drive regulation kind of the PK signal, because PK is potentially downstream of all these different neuromodulators. So yeah, so this is like our team. I think like a team is here, and then all our work is a part of ongoing long-term collaboration with my colleague Tang in Mouse Lab. These are the people kind of from the two labs, kind of we go outing together, and we also owe a lot of things like to our collaborators and also our funding sources. Thank you.
Wonderful. Haining, thank you so much. We have time for some questions. So Chang, please go ahead.
Hi, can you hear me?
Yeah.
Yeah, thank you, Dr. Zhang. So I'm also doing a reward study in cerebellum, but focus on exercise. So I'm just wondering, for the first part of your talk, have you tried to match your data with another model called Anchor? So in their model, they propose that actually it's not about reward prediction error, but rather than the association between reward and the cue.
No, we haven't we haven't done anything along that. I will talk to my student, and then I think like we will. Thank you for the information. We will certainly look towards that. And if you mind, please send me an email afterwards as well. I will we are very new, at least in terms of the lab, we are very new kind of in this field. More like up to a few years ago, like cerebellum is no more than when we prepare kind of for physiological slides. We made the first cut, throw away the cerebellum so that we can mount the cortex and then cut the section, right? So and then so we are really still learning the field, and will love any further discussion and information on this.
Just
Yeah, so
Chang,
sorry.
whose work was it that you said that
Nambudri lab from UCSF.
I see. Okay.
I can send you the papers. So I mean, yeah. So basically, it's pretty similar to reward prediction error, but they did very in detail experimental design to just separate the two different effects. I can send you the paper. I mean, usually you it's hard to I mean it's people just think it's naturally about reward prediction error. So I think it's good to think about in another way. So I mean, I have one additional question. So so your recording was done in the lab hemisphere, right?
Yes.
Yeah, so we did simultaneous recording from left, middle, and right cerebellar cortex. So for my data, it's actually the left and right are pretty distinct, and we see the signals are associating in different, we think different sensory feedback that the animal can sense. Actually, what for our data is actually the right simplex can shows the signal that's more associated with the auditory cue. But yours seems like it's from lab. So I was first wondering, maybe our data shows that the different hemisphere gets different sensory input, but since your experiment also use auditory cue and also shows that the auditory association, but in different hemispheres, so maybe we have to interpret our data differently. So I'm just wondering, so what do you think? Have you tried to record from the right hemisphere?
It's a very good question. We never tried to write. I think like we have only built with the assumption that the two sides are relatively similar. But what I can say is that like from the left, it's very clear that even without any training, you give the auditory kind of stimulation, like there will be this first wave, first phase of complex spikes. So I think like that's always, but certainly like after training and learning, some of the dendrites that previously does not respond become stronger responder. So I think that there is an enhancing component as well. But even naturally, they can.
Thank you.
Sure.
was there another question? Oh, okay.
No.
Haining, thank you so much for taking the time. It was just wonderful.
Yeah, thank you very much. And then like really like I really appreciate all this discussion. And then like if any of you have any further questions or have any discussion or want to collaboration, please really feel free to contact me. And my email is my last name Zhong, ZHONG at OHSU, Oregon Health and Science University, ohsu.edu. Thank you. Oh, I see another hand. Oh, so like thank you. Yeah.
Wonderful.
Okay.
Thank you so much. See you
Yeah.
next. Bye-bye.
Bye-bye.